Hello, welcome back. Today we're going to talk about a special kind of infection that leads to cell transformation and oncogenesis. So let's talk about what transformation is first. So we do an experiment here. We take a hamster embryo and we chop it up and we digest it with trypsin so that we get single cells. And then we plate them out in plastic dishes. And one dish, we simply plate out and watch them grow. And you can see the normal cells growing there, the picture, the drawing on the right, normal. The cells are nicely shaped. They all lay flat on the monolayer. They stop growing when they touch each other. The other set of cells, in the other plate, we've treated with a chemical that is a mutagen. Induces mutations in DNA. And when you look at those cells, you see rare foci, as you can see here, of transformed cells that have different properties. So that's what transformation is. It's giving cells a different property, and one of them is immortality. No, you can't mutagenize yourself and get immortal. Neither can you put bleach in you and prevent COVID, okay? You can't do any of that stuff. And that on the right is a microscope drawing of a transformed cell. You see the cells are piling up on one another. They're not respecting contact inhibition. So that is what a transformed cell is. It has different properties from normal cells. We'll list those properties. But first I want to tell you what happens with cells from different species when you do that. That was a hamster, just to illustrate transformation. It was chemically induced transformation. It turns out that if you plate mouse cells in culture on the top graph, so we're looking at growth on the y-axis with time on the x-axis. This is in days. After a while, after about 30 divisions, so cells in general divide about once a day. After about 30 divisions, the cells die. They undergo crisis. Now, not all of them die, but most of them do, as you can see. We call this crisis. And then a few rare ones begin to divide, and they are immortal. So they have arisen spontaneously without adding a chemical. Those are transformed cells. If you do the same experiment with human cells, though, they will grow for a bit longer, and then they will die. They will not go through this crisis. They will not spontaneously transform. And the reason for that has to do with the ends of the chromosomes. As you know, the ends of chromosomes in cells are particularly fragile because if you lose the ends, which happens during DNA synthesis, the cells will die. That's one of the reasons for aging. Your chromosomes get shorter and shorter and shorter. And in our cells, what keeps us dividing is that we have a structure at the end called the telomerase. And this arises during embryonic development. It's actually a set of proteins. They're called telomeric proteins, including one called TERT, telomere reverse transcriptase. So this is the RT that everyone missed when they were first looking at retroviruses. It's a cellular reverse transcriptase. And during embryonic growth, this telomerase adds sequence to the ends of chromosomes, kind of like a buffer. And then when you're born, these are usually inactive, these telomerases. And that's why with time, the chromosomes get shorter and shorter. And so you see they're degraded because we have the 5' end problem. And so they're degraded unless they're long as made by telomerase. In culture, in cells in culture, the telomeres shorten at each cell division about 4,000 bases. So that's why our human cells only last about 50 divisions. The telomeres shorten and the cells die. But mouse telomeres are longer. That's why they can grow longer and proliferate more generations, which increases the likelihood of a spontaneous mutation, right? At every cycle, the DNA is mutated by the error-prone polymerase. And so you just need the right mutation to make the cells immortal. So that's the relationship between the ends of the chromosome and immortality. So if you took the telomerase gene, TERT, and put it in a cell, you could make it immortal because it would keep the ends of the chromosome. That's one way that you get immortality. But there are other ways as well, as you'll see today, of telomeres in cancer. So we're just talking about immortality so far. But again, with cell division, the telomeres shorten, and when they get to a certain length, the cell division stops and the cells die. With some cancer cells, the telomere length remains the same because the telomerase is active. So you can imagine that a random mutation could activate the telomerase, and then cells would keep dividing, and eventually they'd make a tumor. So that's dividing forever is immortality. That is transformation. When cells divide enough, they sustain mutations randomly, and eventually they will become a tumor. So transformation is not enough for making a tumor. You have to have more mutations, usually about 12 in different kinds of proteins, different genes encoding different proteins. So you start with transformation, and then that leads to a tumor or cancer. So here are some properties of transformed cells. They're immortal, they grow indefinitely, and HeLa cells, which we'll talk about today, are an example of immortal cells. They're named after Henrietta Lacks, from whom they were taken. They have a loss of anchorage dependence. They don't actually need a plastic to grow on. They can grow floating. They lose contact inhibition, which means normal cells, when they touch each other, they stop growing, and transformed cells just keep piling on top of one another, as you can see in B. They will make colonies in semi-solid media. What does that mean? So instead of putting liquid media on a cell culture, you put an agar overlay. You can embed cells in the agar, and they will grow if they're transformed. But if they're untransformed and if they're cells by themselves, they don't like to grow. They need neighbors. You'll see why that is today. And they also have decreased requirements for growth factors. Serum, when you put serum on cells in culture, you usually need 10% serum to get cells to grow. That is supplying growth factors. And transformed cells need less growth factors than, say, primary cells that you make, say, from the inside of your cheek or from foreskin. Foreskin fibroblasts that you make all the time from, you can get them in the hospital, you can chop them up with trypsin and make primary cells. They only grow for a certain amount of time because the ends of the telomeres shorten. You know, foreskin is made from a newborn, so it's got the full ends of the chromosome, but every doubling in cell culture, it decreases 4,000 bases, so rapidly you lose the ends of the chromosome. Okay, so oncogenesis is different. Oncogenesis is the development of cancer. Right? Transformation is just giving those properties to cells. But if you take HeLa cells and inject them into a wild-type mouse with an intact immune system, they'll be cleared. So they're not cancer cells yet. And of course, the tumor is swelling caused by abnormal growth. These cells just keep proliferating uncontrollably, and eventually they make a tumor, which could be benign and remains restricted, or cells can break off and spread elsewhere and make growths elsewhere, and that's where cancer is a killer, of course. Cancer is a genetic disease. It involves mutations in our DNA. There are about 8 million deaths per year in developed countries caused by cancer. What about the other countries? Hard to know, right? We don't have good reporting from many countries, so many more obviously are likely. And about 12 mutations are needed to make a cell into a cancer, and these are in cell signal transduction pathways that affect cell growth, cell survival, determination of cell fate, maintenance of genome integrity. So, you know, there are enzymes in us that repair breaks in DNA, and if they don't work, then the DNA is mutated and it can grow into a cancer and so forth. And these mutations, you can get from your parents. You can get them by DNA damage, which happens to us all the time. Environmental carcinogens, you know, the air in New York is very nice, but it's still full of pollutants, which give a higher rate of respiratory cancers than other parts of the country. And viruses can do it as well. And, you know, depending on how many mutations you inherit from your parents will depend on how long it takes for you to develop a cancer. If you inherit just a few, it will take longer. if you inherit more, you will have a childhood cancer. And you know, humans live longer now than they ever did. The average lifespan, as we'll see in the vaccine lecture, has gone from 35 years of age on average to over 80. And our bodies are not made to be 80 years old. That's the effect of public health and medicine and so forth. All we are made for, all we evolved for, is to reproduce, which can be done by the time you're 30, and everything after that is gravy. You're not protected against cancer. So as you spend a lot of time on Earth, you get more and more mutations in your genome, you get cancers, you get dementias, and many other unfortunate kinds of diseases. Okay, but some people do live over 100, amazingly, and they would be interesting to study to see what it is in their genome that they actually have. So transformation and oncogenesis are two different things. Transformation is not a cancer. Transformation is simply making cells have different properties in cell culture, including immortality. And oncogenesis, the formation of a tumor. So there's a mouse where you've inserted cells into it. You can see on its back there's a tumor. It requires additional genetic changes. And the point of today's lecture is that by studying cells transformed by viruses, we can understand the events that give rise to cancers. And it gives a very nice window on that, which we didn't have before. Important thing to remember is that a virus doesn't cause cancer. A virus transforms cells, and then the cells keep growing and the mutations accumulate, and then the cell becomes tumorigenic. And even for, as we will see later, HIV causes immune cells to proliferate continuously because they're responding to the presence of virus. And that proliferation eventually makes those cells turn into cancer. So the virus is not causing the cancer. It is a trigger for sure, but it doesn't do it. So here's a list of the human cancer viruses. About 20% of human cancers are caused by viruses, including RNA and DNA viruses, HTLV1, HIV, hepatitis C virus, or the RNA viruses, DNA virus, Epstein-Barr, Kaposi sarcoma, herpes virus, hepatitis B, human papilloma, and Merkel cell papilloma. We'll mention human papillomaviruses today. These are viruses that cause warts, of course. There are hundreds of wart-causing papillomaviruses, and a few of them are at risk of giving you different cancers, as you can see, cervical, penile, anogenital, head and neck cancers, and we vaccinate against those viruses for that reason. We'll talk about the mechanism today, but the others we will not talk about. We're going to talk about retroviruses today, which taught us how viruses can trigger cancers to form. And these viruses transform cells, for the most part, in different ways from the retroviruses that we're going to talk about today. The papillomaviruses are similar, and Merkel cell polyomaviruses, but all the others are different from what we're going to talk about today. But as I said, it's instructive to see how these work. Now, many years ago in this course, I used to say transformation and oncogenesis is not required for the replication of any virus. I said that for years, and then one year I got an email from a virologist somewhere in the US, and she said, I study walleye dermal sarcoma viruses that causes tumors in fish, and the tumor is essential for the reproduction of the virus. It's the only example that we know of. So now I put an asterisk in there, and you know, you're too young to know what the asterisk means, but when you have an exception, there's always an asterisk. Walleye dermal sarcoma virus is an RNA tumor virus that causes tumors of fish, as you can see. And those tumors fall into the water and they spread the virus. If it weren't for a tumor, the virus wouldn't be able to spread. So it's one example of how transformation and oncogenesis is required for the transmission of this particular virus. All right, so the story I want to tell you today begins in 1909. Peyton Rouse is an MD working at the Rockefeller Medical Institute, which is what Rockefeller University used to be called. I point to my right. I have this general feeling that that's downtown, but I don't know if it actually is. Chickens for years were known to get tumors, sarcomas, solid tumors. Here's an example of a solid tumor. And one day, the story goes, a farmer brought him a chicken and Rouse cut the tumor off. Can you imagine a farmer bringing a chicken to a lab nowadays? I wonder if it was alive. He cut the tumor off and chopped it up, filtered it through a filter so the viruses would pass through. So by 1909, we know what viruses are, right? He takes the filtrate and he injects it into another chicken, and the chicken develops the same kind of tumor. Cancer can be caused by a virus. Oh my gosh, nobody thought that this could happen. This is very early on in the history of viruses. Well, it turns out in 1908, a year before, this same experiment had been done with a leukemia virus of chickens, but people didn't think that leukemias were cancers at that time. They thought a cancer is a solid tumor. And of course, leukemias are cancers. And eventually, so that 1908 experiment was the first one. It took 50 years for people to believe Rouse that he was right. 50 years. He got the Nobel Prize in 1966, died shortly afterwards. But he actually stopped working on it himself because no one believed him. But eventually, with time, people figured it out. And I'm going to tell you that story today. And his virus was called Rouse sarcoma virus. It led to 2 other Nobel Prizes in this story of how viruses cause cancers. And I want to quote from this book. Has anyone read The Emperor of All Maladies? Yeah, it's a great book. 2 people. You should read it. I know you don't have a lot of time, but just great. The writing is great. The science is spot on. By the 1950s, cancer researchers had split into 3 feuding camps. This is perfect. The virologists, led by Rouse, claimed that viruses cause cancer, although no such virus had been found in humans. Epidemiologists argued that chemicals caused cancer, but they couldn't say why. And the third camp said there's some gene in us that's causing cancer, but we don't know why. So they're all right in the end, right? But there are 3 different camps feuding. Now, Howard Temin comes along. We've already mentioned him. He co-discovered reverse transcriptase in retrovirus with David Baltimore. In 1951, though, he's a young scientist. He goes to Caltech to study with Renato Del Becco up there. He's a scientist who eventually makes the plaque assay for animal viruses. But he goes to study fruit flies, and he gets bored with fruit flies. So he decides to study viruses. And what Del Becco is working on is Rouse sarcoma virus, right? So this is the virus Rouse had identified in 1909. People are still trying to figure out how it causes cancer. And Del Becco's lab was known for being cutting edge. So he developed the plaque assay right away because he knew the quantitative assay of infectivity would be important. And he also knew that cell cultures could be important at figuring out how cancer works. And so up until this point, people took Rouse sarcoma virus and injected it into chickens, and it caused the tumor. But how much biochemistry can you do with a chicken? Not a lot, but you can in cultured cells. And so that's what Temin and Del Becco envisioned. Somehow reproducing, they said cancer, but it's not really a cancer. It's transformed cells in a petri dish. And so here's another quote from Mukherjee. He added Rouse sarcoma virus to a layer of normal cells in a petri dish. The infection incited them to grow uncontrollably, forcing them to form tiny heaps containing hundreds of cells that Temin called foci, which he reasoned represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably, pathological mitosis. So I would argue it's not a cancer. It's a transformed cell, but it's the beginning. So he was on the right track. And so here are some of his cells that he saw. He infects avian cells, right? Because these are chicken, this is a chicken virus. And here they are in culture. And there you can see a focus, one focus of transformed cells. They happen to have a fusiform shape. And then there's another experiment. The cells become round and refractal. They pile up on one another. So virus can do this. The same thing when cells get immortal, a virus can do that as well. This was just brilliant because this led to figuring out what was going on. So Temin goes on, of course, to discover reverse transcriptase because these cells are permanently changed. They're transformed permanently. They don't revert. So Temin said, and this is what's the best part of science, he reasoned, well, if the cells are permanently transformed, something had to happen to their DNA. And tumor viruses are not DNA viruses, they're RNA viruses. So somehow they have to convert their RNA to DNA. So that's the beauty of the logic, right? And then he said there's got to be an enzyme there, and he in Baltimore eventually found it, as I've told you before. So that is where we are so far with RNA tumor viruses. We're going to go further, but first I want to tell you that DNA tumor viruses also play a role. So at some point, 1962, people find that if you infect cells with a polyomavirus, so think SV40, you can get transformation of baby hamster kidney cells, BHK. And in 1964, infection of mouse cells with SV40, you can also get transformation. So most of the cells in these experiments die because they are infected with the virus and it goes through its lytic cycle. But some of them don't die, they grow forever, they're transformed. So now we have RNA tumor viruses and DNA tumor viruses, both transforming cells. So now we have to figure out how. So how can a viral infection transform a cell? Three things. Cytopathic effects have to be reduced or eliminated, right? The cell can't die to be immortal, right? That's just totally makes sense. Second, you have to reduce viral replication. In fact, the observation is that transformed cells do not make virus particles. They don't. They must have lost most of the machinery to make virus particles. And third, the cell has to continue to divide, so it becomes immortal. And so the little bell there means, does this ring a bell? Yeah, it's a kind of persistent infection, right? Where the genome is always there, or at least part of it. No particles are produced, and the cells are always containing the genome. Okay, so let's see if that makes sense. Our first question is, which of the following is not a property of transformed cells? Increased requirements for growth factors, immortality, loss of anchorage dependence, loss of contact inhibition, colony formation in semi-solid media, which is not a property. All right, let's see how we did. That was an easy one, right?
Always that one guy.
It's always one guy or gal. It could be either one. Colony formation in semi-solid media is a property. I'll show you the slide. I know, it's hard to remember all this stuff. Where's the slide? Yeah, that would be early on. Properties of transformed cells. Colony formation in, you can't see it, yeah, sorry. Colony formation in semi-solid media. So that is a property of transformed cells. The one that is not is increased requirement for growth factors, decreased requirements for growth factors, okay? All right, so let's talk about RNA tumor viruses first. So the way cancer was understood is to get an understanding for the theory of cell growth control, because cancer is about cells growing uncontrollably. And so we have people studying retroviruses and RSV, and then we have people studying DNA viruses, like polyomaviruses and adenoviruses, and then we have people studying cancer cells in vitro, and they all converge in the 60s and 70s to give us our current understanding of cancer. So we're going to start with the retroviruses, which of course begins with Rous sarcoma virus, but rapidly grows after Temin's finding to include other viruses as well. So what you need to know something about chickens. Every chicken you eat has retroviruses in it, okay? But they're not going to harm you. And you can eat the blue eggs too. Those retroviruses are not going to harm you. Avian leukosis. Leukosis is another word for leukemia. Avian leukemia viruses are endemic in all chicken flocks around the world. That virus that causes leukemia in chicken was identified in 1908 by infecting chickens with a cell-free filtrate. These chickens had leukemia. Most chickens are infected with the virus, ALV, within a few months of hatching, no matter where they are. And leukemia occurs sporadically, important word, which you'll see why in a minute, occurs sporadically in infected birds that are over 14 weeks of age. About 3% of those birds develop sporadic leukemia. And the rest of the birds, 97%, they're all infected. 97% have viremia. They get immune. They don't get leukemia. And those are the birds that go on to be eaten and so forth. So every bird has this virus in it. So you're starting to think now, is it having to do with Rouse? Yeah, it does have something to do with Rouse. These birds, if they're allowed to age, if they're not killed early on, if they're allowed to age, they develop other cancers. After having this virus infection, they develop other cancers, including sarcomas like the one that Rouse was studying. And if you take Rouse sarcoma virus and infect a chicken, you get a solid tumor. You don't get a leukemia. The virus starts out as a leukemia virus, but something changes to make it cause sarcomas. And that's what Rouse isolated. Rouse sarcoma virus is one of these viruses from an aged chicken that had previously been infected with avian leukosis virus, and it caused a sarcoma. And he did this multiple times, and other people, of course, tried to repeat his results. And most of the time, the viruses that people got from these tumors were defective. They were missing viral genes, so they could not reproduce on their own. Rouse was very lucky. He identified a replication-competent virus. All right, so that's 1909. Fast forward, so we go past Howard Temin in the 50s, who figures out you can transform cells in culture with this virus. So then other people get interested in working on it. And two people here, Michael Bishop and Harold Varmus, they're studying cells transformed by Rouse sarcoma virus, and they're isolating virus from these cells, and they see that the viruses are recombinants. They have a piece of the viral genome replaced with a cellular gene. And in fact, Rouse sarcoma virus is nothing more than avian leukosis virus with a segment of host DNA added. And that segment of host DNA we call an oncogene because it transforms cells and eventually they become tumors. So they identified the cellular oncogene in Rouse sarcoma virus in 1976, working at UCSF, and they called it V-SARC, SARC for sarcoma, and V, the viral version, because there's a C-SARC, there's a cellular version as well. And in 1989, they both got the Nobel Prize for that discovery. Harold Varmus used to be head of NIH for 10 years in the Clinton years. Now he's president of Sloan Kettering. And I interviewed both of them separately. So if you want to hear them, the TWIV is Harold Varmus and the YouTube is Mike Bishop. Very interesting to hear their stories. Yeah.
Professor Varmus teaching here. He's teaching a class.
He's teaching a class?
Science and Society.
Science and Society. Okay. That must be good. Did you take it?
Yeah, I took it not this past one.
Oh, he has a good book. He's a very eloquent speaker and writer and very smart guy. But you know what? He majored in comparative literature in college, or either that or poetry. I don't remember which, but in the interview he said, I rapidly realized I couldn't make a living because I wasn't that good a poet.
He went to a PhD program in English.
In a PhD program, and then he said, I'll be a doctor, so. And then he became a really good scientist. He went to Mike Bishop. And it's a great story. He talks about it in TWIV. He just wandered into Mike Bishop's lab one day and said, what are you doing? Can I work with you? It was very different back then. Okay, so the point is here, can you imagine someone wandering in? They're like, what are you doing here? Get out of here. So that's the insight they provided, right? All birds get infected with ALV. Some of them get different kinds of tumors. The tumors are caused by viruses derived from ALV. Most of them are defective, but they're also different. They pick up different oncogenes and cause different kinds of tumors. Rouse's tumor was a sarcoma, and his virus was not defective. He was very lucky. But each of these viruses that causes a different tumor is ALV with a different gene picked up from the host cell. And I say here, it's a gold mine for molecular oncology. Oh my gosh, all these viruses that spontaneously arise and cause different kinds of tumors in chickens, you just identify the cell gene, and now you get insight onto what can cause immortality. So people did this for many years. And so these are the genomes of what we call transducing retroviruses. So transduction, remember, is when a virus infects a cell, picks up a piece of host cell DNA, and then delivers it to another cell. That's a word that was first coined for bacteriophages, and now we know animal viruses do it. So avian leukosis virus infects a cell, picks up a cellular gene, and then brings it to another cell. That's transduction. So these are transducing retroviruses. On the left, we have all the different avian transducing retroviruses. So these are infecting chickens with, well, you don't have to infect them. They're naturally infected with avian leukosis virus. And then identifying different kinds of tumors and the viruses that infect them, and then the oncogenes that are picked up. So here is avian leukosis virus. It's a typical retroviral genome, GAG, Pol, envelope genes. And then look, Rous sarcoma virus has the Sark gene from the cell inserted in addition to these three. So it's not defective, can still reproduce. And so that's a Rous sarcoma. Then you have a myeloblastosis virus where the oncogene is called MIB, and that now has disrupted the envelope gene, so the virus is defective. So you may say, how do you grow these? Well, you need to have the wild type virus in to provide envelope. And then these are all different kinds of cancers and different oncogenes that have been identified that are responsible for the transformed cell. So each of these genes, when expressed in the cell via virus infection, causes the cell to divide uncontrollably. Most of those oncogenes actually have additional changes compared to the cellular version, which probably facilitate a transformation. Now on the right, people did the same kind of experiments with mammals. So that's a murine leukemia virus, GAG Pol envelope. They infect mice and they identify different kinds of cancers, leukemias, sarcomas, and they identify the retroviruses and the cellular oncogene that has been picked up. And you can see these viruses are defective as well. So you can now say, we have all these cellular genes, which when picked up by a virus and inserted into another cell cause transformation. So I want to make clear what I'm talking about by defective versus non-defective, all right? Defective virus is missing some gene. So here's the avian leukosis virus, and Rous is not missing any. We have GAG, Pol, and envelope. Sark is additionally added, but avian myeloblastosis virus BA1, the MIB has stuck right into the envelope, so there's no envelope gene, and the virus can't reproduce, so it's defective. And these are typically missing envelope proteins, and the genes are deleted in the process of putting the oncogene into the viral genome. You need to have the virus, the helper virus, which in this case would be ALV, in order to get AMV to replicate. So if you identify avian myeloblastosis virus in a tumor, if you want to grow it in cells, you need to co-infect with ALV, which will provide the envelope. So how do the viruses capture the oncogene? There are two mechanisms here. I just wanted to show you this in case you're curious about it. So remember, the retroviral genome is reverse transcribed into a DNA, shown circularized here, and then it integrates into host DNA. And so if the integration is near an oncogene, here the oncogene is in red, like Sark, right? If it integrates next to the oncogene, then in some cases there can be deletion of the right-hand LTR, so DNA rearrangements occur all the time. So if you delete that LTR, it has to stop the transcriptional stop signal. So now the mRNA synthesis will include the oncogene because the LTR is gone, there's no stop signal, and the left-hand LTR, which supplies the promoter, now makes an mRNA of GAG, Pol, and in this case, the oncogene. And then that mRNA will get incorporated into a virus particle, and eventually that virus will deliver the oncogene into another cell where it's integrated into the DNA. So that's one mechanism. The other, there's a slightly different mechanism on the other side. But the point is, you have integration next to an oncogene and then production of a long transcript. So these cell genes that get captured by viruses to transform cells, they're called proto-oncogenes. There are over 60 that have been identified so far. And in all the cells where they've been studied, they control cell growth, cell division, right? Cells don't divide all the time. And so these genes are involved in that regulation. These cellular genes have a C in front of them, C-SARC, MYC, C-MOS, et cetera. So C-ONCs. And the virus versions are called V, V-SARC, V-MYC, et cetera. These oncogenes, so there's a cell cycle on the upper right, which you remember has a number of phases. It has a mitosis phase shown in red. Then it has a G1 phase. It has a S phase, a synthetic phase where the DNA is duplicated. And then a growth phase, the cell gets bigger, and then it divides in two. And that happens roughly every 24 hours. The proto-oncogenes, all these genes give a go signal into mitosis. They say, okay, you can divide, and we'll provide the resources for that. So you can see why putting these genes unregulated in a virus could cause transformation. Now, when people began to study these proteins in detail, these oncoproteins, all of a sudden it turns out they're all involved in the growth control pathway. So what is the growth control pathway? In order for a cell to divide, you need a number of steps shown here on the right. First, a growth factor has to bind a receptor on the cell surface. If there aren't growth factors present, the cell won't divide. It means that the environment is not rich enough for division. And then there are receptor protein tyrosine kinase that become active and involve other proteins by phosphorylating them. And eventually, these proteins go in the nucleus and turn on the transcription of genes that will make the cell divide. DNA polymerases and other genes as well. So that's cell growth control. It's highly regulated. Each of these components is highly regulated so that you don't have cells dividing when they shouldn't be. Why? Because they would become transformed if that happened. Now, each of these proteins is encoded by an oncogene. So here we have, let's find SARC. SARC is up here. This is feline sarcoma virus. This is Rouse sarcoma virus, right? These are non-receptor tyrosine kinases. They're also receptor tyrosine kinases. So the feline and the avian SARCs can be this protein here. Rouse is this particular protein here. And all these other oncogenes, you can see they're even growth factors that are oncogenes. There are receptor protein tyrosine kinases. There are hormone receptors. There are non-receptor tyrosine kinases, G proteins, et cetera. Even transcriptional regulators are captured as oncogenes by transforming retroviruses. So you can see that the study of these viruses totally illuminated how cell division is controlled. We had very little clue about it before then, and it was all sorted out in like 10 years by studying these. It's just a brilliant period in molecular biology. So the cell cycle, again, is controlled by these oncogenes, the products of these oncogenes. And we understood that by studying these transforming retroviruses. These are dominant oncogenes. You have one amino acid change in one copy. So you have two genes in each cell. An amino acid change in one copy is enough to make it into an oncogene and make the cell keep dividing. And so retroviruses that pick up these oncogenes transform cells by three ways. The Rouse mechanism is rapid. You inject the virus into the bird, you get a tumor in about 2 weeks. That's because Rouse sarcoma virus has an oncogene in its genome and it delivers it to the cell. The protein is made as soon as replication occurs, and that turns on cell division. And of course, so you start dividing uncontrollably, you accumulate mutations in the gene, and eventually you get 12 or so that lead to a cancer. So you see, the virus is just making the cells divide. It's not making the actual cancer. Then we have viruses with intermediate kinetics like ALV, right? I said this takes a little bit longer, months. There's no oncogene in ALV, right? It's the parent of all these oncogene carrying viruses, but it can still cause a cancer, which is leukemia. And that's because when it integrates next to an oncogene, it turns on its transcription. And then finally, you have viruses with very, very slow kinetics of tumor formation. It can take years. HTLV, human T lymphotropic viruses. There's no oncogene. It does not integrate next to an oncogene, but it makes a regulatory protein, a transcriptional regulatory protein that's needed for transcription of viral genes that happens to activate the promoters for oncogenes. And that's how it it causes transformation. And so these are shown here in terms of the genomes. Here on the top is a rapidly transducing retrovirus. That's Raus, which is carrying an oncogene, integrates into the genome, and the oncogene is produced, transforms the cell. Here are the cis-activating like ASV. They don't have an oncogene. They integrate next to an oncogene, and transcription from the right-hand LTR produces an oncogene mRNA uncontrollably because the provirus is not regulated. Its promoters are on all the time. It wants to make a lot of virus. And here are the transactivating oncogene retroviruses. They have no oncogene. They don't integrate next to a oncogene, but they make here X protein, which is a transcriptional activator that you need for activity of the LTR promoter. And that will turn on cell genes, including here, in this case, IL-2 and its receptor. And that makes T cells proliferate uncontrollably. IL-2 is a growth factor for T cells, and they keep dividing and dividing, and eventually they become leukemic. So back to wall-eye dermal sarcoma viruses, the mammalian transforming retroviruses that we've talked about, they transform cells as a mistake because they have to integrate their DNA. And it doesn't matter really where they integrate. If it happens to be next to an oncogene, then it gets turned on, but it has no implication for the viral life cycle. These viruses do not need to transform cells in order to reproduce, with the exception of wall-eye dermal sarcoma virus. So there's no requirement for the virus for transformation or oncogenesis. It's an accident of the way the virus reproduces, which is by inserting into the DNA. Okay, let's see how we do now. Which of the following allows Raus sarcoma virus to transform cells? Presence of the envelope, presence of a pol, presence of SARC, presence of LTRs, none of the above. Yeah. So the right answer is the pol gene. I mean, that's the best answer, right? That's what transforms cells. Now, I know envelope you need to infect a cell, but it doesn't transform, right? It just allows you to infect. Yeah, you need the LTRs, but if you don't have a SARC, even with LTRs, you don't get transformation. So that's why you need the SARC gene. Let's talk about DNA tumor viruses. These also contributed to our understanding of cancers. So in the 1920s, these are identified, and we start to do molecular studies like we have just talked about with RNA tumor viruses. And this starts with the first oncogenic DNA tumor virus. That is, infection leads to tumors, but you know better than that. It leads first to transformation. This is a papillomavirus that causes warts in rabbits. This was discovered by Richard Shope in 1933. So these are rabbits, and they have warts on their faces. They don't hurt the rabbits. They fall off eventually. They just look a little angry. But they're caused by, so these are the same kind of viruses that cause warts on us and which can lead to certain cancers. These are benign warts. And people see these and they think they're a cross between a jackrabbit and an antelope because they have horns. So they call them jackalopes, right? But there's no such thing as a jackalope. There's this. However, what I thought was cool, last year, this week, so my son is an amateur DJ here in the city, And he played a concert called Feast of the Jackalope, which this is, there's a jackalope with both ears and horns. And this is his name, by the way, because it's like HeLa cells. I taught him that Henrietta Lacks made, and so he's Aidan Racaniello, so he's Ira. So I thought that was really cool. They didn't do it again this year, so I used last year's. And I said, this is not a thing. He said, well, it's art, it doesn't matter. So in 1953, Ludwig Gross there discovered murine polyomaviruses, so now mice instead of rabbits. He can infect the mice and they cause rare tumors. So these viruses, the natural host is the mouse, and they're found in many mice, but they don't cause cancers in mice. They make tumors in many tissues of rats, hamsters, and rabbits, different species. So he called them polyoma because many different kinds of tumors. I really like this picture because all of his cages are made of wood, the mouse cages. Somebody made those. Nowadays, they're all molded plastic, right? But I think that's kind of interesting. Another DNA tumor virus discovered by Bernice Eddy and Maurice Hilleman in 1962, SV40. This virus causes tumors in hamsters. So the natural host is a monkey, a non-human primate. So it's called simian virus number 40. It doesn't cause any tumors in monkeys, causes tumors in other species. It doesn't even transform monkey cells in culture. By the way the polio vaccines are made is you take a non-human primate and you sacrifice it and remove its kidneys and make cell cultures, and then you infect with poliovirus and you make a lot of vaccine. And it turns out that monkeys, their kidneys are infected with SV40. So millions of Americans were inoculated with SV40, both in the inactivated polio vaccine and in the infectious polio vaccine in 1960. As far as we can tell, it hasn't caused any tumors in humans, but lawyers went crazy for years over this, suing and trying to prove that this caused someone's cancer. But it was an accident, of course. We didn't know. We should have known. We should have thought that there would be other viruses there, but we simply didn't. And so here are how cells respond to infection with SV40 and mouse polyomavirus. So SV40 is a virus from non-human primates. It is in monkey cells. Monkey cells are permissive for SV40. In other words, the virus binds and reproduces and kills the cells. The mouse polyomavirus, however, the monkey cells are non-permissive, so they don't even replicate at all. In the mouse, SV40 is non-permissive. Mouse polyomavirus is, again, permissive. It's the native host. Hamsters are semi-permissive for both viruses, and rats are semi-permissive. And the asterisk means in those species, these viruses cause tumors. Semi-permissive means you get to DNA replication and you stop. So you get all the early gene expression, right? And then you get to DNA replication and you don't do it for whatever. And that's semi-permissive, but the virus is making early gene product. Now, this transformation can be done in cell culture. You don't need to infect animals to get tumors. You can infect cells with polyoma or SV40. You get rare transformed foci. And we'd like to know, why is it rare? And how does it relate to tumor formation in animals? So this is transformation in culture cells. Is there a relationship with what we're seeing in animals? Another DNA virus found to cause tumors are adenoviruses. There are many human serotypes, but they do not cause tumors in humans. We are the natural host for adenoviruses. But some of the serotypes, 12 to 18, cause tumors in hamsters. Again, a pattern emerging, the wrong species. And some are, and other serotypes are also tumorigenic in hamsters. Again, the tumors and the transformation, just like the polyoma viruses, which SV40, the papilloma, the rabbit papilloma virus, they're rare events. So why is this? We're going to answer this. A key finding when people were studying this in cells in culture is that in transformed cells, they all produce T antigens. Also in animals, if you have a tumor in a mouse or a hamster or a rabbit, you find viral T antigens always. For SV40, that's the T antigen we've talked about a lot. There are also T antigens for polyoma. For papillomaviruses, they're encoded by the E6 and E7 genes, but we'll call them T antigens. And for adenoviruses, right, they're E1A and E1B. And E1A we've talked about as involving something to do with RB, and we'll see that in a moment. These are all different proteins. So the SV40 polyoma, papilloma, adeno, T antigens are all different, completely different proteins, but they're all present in transformed cells and in tumors. So that must be telling us something, right? Remember, the T antigens are essential. You need them for replication. They activate, remember, they recruit the DNA synthetic machinery to the cell. They turn on cell division, right? To get the DNA synthetic apparatus going. They also are involved in transcription. They're needed for viral DNA synthesis. And they are the only viral genes always found in transformed cells or in tumors. You can find a few others here and there, but the only ones that are consistently present are T antigens. And if you take the gene for a T antigen and put it in cells in culture, you can make them immortal. So we used to do that in the lab all the time. We would take a cell. You can take a swab and take some of your cheek cells, put them in a dish. You can look at them. You get cells just by swabbing your cheek. They'll die within 50 divisions. But if you put T antigen in them, they're immortal. So I could make a virus cell line if I wanted to. Just T antigen will do that. And then people were doing more experiments to figure out what is with the T antigens? Why are they there? First, they found that T antigen binds a cell protein called P53. This was the discovery of P53, which is a huge protein in oncogenesis now. Secondly, the early genes of adenovirus, the E2 genes, require a cell transcription protein, a member of the E2F family of transcription factors, and E2F binds RB. And all these proteins, P53, RB, and E2F, are all part of the normal cell cycle control pathway that we're going to see again. We've already talked about it a little. So here again is the cell cycle where 24-hour cycle, mitosis is expanded at the top, but really what you need to know is that there's one period of cell growth, there's DNA replication, and then preparation for cell division. So when you stimulate a cell to divide by, say, a retroviral oncoprotein, the cell proceeds through this cycle. They enter G1 into S and eventually back to mitosis. But there is a restriction point down here that will stop things if conditions are not right. So we have one control decision up here at the top, which is controlled by these oncogenes, right? The oncogenes that were identified in RNA tumor viruses. And they are saying basically, are there growth factors in the medium that we need to divide? And if so, they start the signaling, which pushes the cell through division. But there's another control point there shown in red in G1. If conditions in the cell are not right, then we have a restriction at that point, and the cell will not go beyond partway through G1. So you get no DNA synthesis, no cell division. And this protein that regulates this step is called RB, which we have encountered before. So let's talk about it a little bit more. This is called retinoblastoma protein. If you lose both copies of the gene, you develop retinal tumors of the retinoblasts, where this protein is found in the retina. These cells are typically gone by age 5, so you see retinoblastoma tumors mostly in kids under 5 years of age. But they were studying these tumors and found that this protein was missing. And why am I telling you? Because it interacts with T antigens. That must mean something, right? This is a recessive oncogene. Both copies have to be lost to lose this restriction point. If you delete both copies of RB, then the cell divides forever and you develop these retinal tumors. P53 and RB are implicated in many human tumors. They're mutated in many human tumors. So here's the mechanism of action. So we have our cell cycle control pathway here, which all these components identified by studying transducing retroviruses. We have the receptor tyrosine kinases, we have the growth factors that bind these and stimulate a signaling pathway that then basically activates cell division. It causes phosphorylation of RB, which then turns on transcription of genes involved in the G1S transition. And the RB portion is shown on the right. Remember, RB is usually bound to E2Fs, and E2Fs are the transcription factors that are needed to make all the proteins that make a cell divide, like the cyclins and so forth. And in a cell, if you have a growth factor binding, eventually RB is phosphorylated, it pops off of E2F, and then E2F can activate genes involved, say, in DNA synthesis and so forth. So that in a normal cell, that is what happens. RB is phosphorylated. But as you know, certain DNA viruses need to make cells divide so that they can replicate their DNA. We talked about this some time ago. And how do they do that? With T antigens. And we talked about this before. T antigens bind RB and they pop it off E2F. So it's the same effect of this phosphorylation of RB, except it's done by a viral protein, which needs to make sure once it infects the cell that the cell is dividing, so the cell is making DNA polymerase and the viral genome can be replicated. Right, so that's it. And here is shown that mechanism. So again, we have E2F bound up by RB. E2F again is the transcription factor you need to turn on genes to get cells to divide. It can be phosphorylated during normal growth to pop it off of, RB can be phosphorylated to pop it off E2F. Or if it's a virus-infected cell, making E1A or large T or E7 from the papillomaviruses, that will bind RB and pop it off E2F, and that gets the cells dividing. So you can now start to see why these T antigens are present in every tumor, right? Because they are inactivating RB and making the cells divide forever. There's one more control step that we need, though. When cells start to do unscheduled DNA synthesis, like virus-infected cells, then there are proteins in the cell that say, this is not happening. And when you replicate DNA, you often make DNA breaks. Double-stranded DNA breaks can be sensed by a pathway that ultimately leads to activation of P53, that one protein, that other protein that we said binds T antigens. P53 is basically sensing DNA damage or unscheduled DNA synthesis. And if it catches it, it will turn on pathways that lead to cell cycle arrest and apoptosis. So obviously viruses have to counter it, because if they go in a cell, make a T antigen, and get the cell to start dividing, P53 is going to say, no, this is not scheduled. And how it does that is more complicated than we have time to talk about. And it will shut down the cell and kill it. So viruses have to counter it. And viruses counter P53, no question about it. And what proteins do you think counter P53? Yeah, some of the T antigens do as well. So here's P53, which basically senses DNA replication and leads to apoptosis. So let's see, we have here the E6 protein of papillomaviruses, which causes degradation of P53. The large T of SV40 sequesters P53, so it can no longer sense unscheduled DNA replication. E1B of adenovirus is a protein that binds to P53 and activates it as well. So remember, E1A was the protein that frees up RB so that E2F can be available. E1B, its counterpart, will take care of P53. So basically, this is how the DNA tumor viruses get cells to divide forever. Although there are a few questions we need to answer yet, and we'll do that after this last question. So T antigens are encoded by viral genes that are essential for replication, present in tumors and transformed cells, encoded by viral genes that have been incorporated into the cell genome, antagonists of cell cycle checkpoint proteins, all of the above. All right, let's have a look.
Yes.
Hey, that surprised me. It's taken a bit of time, you know. Yeah, so all of these are correct. They're encoded by essential viral genes. They're present in tumors and transformed cells. They're encoded by genes that have been incorporated into the cell genome, and they're antagonists of cell cycle checkpoint proteins. Okay, so let's finish up and understand how all this is working. Two things. Why are all genes except T antigen deleted or turned off in SV40 polyoma and adenovirus infected cells, transformed cells? Remember, I told you that only the T antigens are present in all of them, tumors and transformed cells. Why are the other genes not present? And why is transformation rare? Let's answer those questions. So transformation is rare because there are two low probability events that have to happen before transformation. First, the late genes need to not be expressed because those encode capsid proteins that typically can lyse the cell. And so you depend on rare spontaneous deletions of late genes. So a virus infects a cell, DNA rearrangements occur. Sometimes the late genes are deleted and that rare virus no longer is going to kill the cell. Or, now that's a case where you're infecting a permissive cell. If you infect the wrong species, you never get into the late genes. Remember, semi-permissive means up to the point of DNA synthesis. So you don't have late gene expression, so you're not going to kill the cell. So if you take a monkey virus and infect a hamster cell, it's semi-permissive, you don't kill the cell. See, that's why using the wrong host was a good move. And secondly, T antigen has to be on. It has to be on all the time and transmitted to every cell in order to keep the cells dividing. So how do you achieve that? You integrate T antigen DNA into the host DNA, and that's a very rare event as well. So the DNA is in the nucleus. Occasionally, it will integrate into the host cell. So you have these two rare events happening, and that's why transformation and tumor formation are rare. And these events, again, just like the RNA tumor viruses, they're abnormal epigenetic processes. They're not needed for normal virus reproduction. They're not needed for transmission. Because in the natural hosts, these viruses replicate and kill the cells and transmit. They don't cause tumors. It's only when we infected the wrong host did we discover this. So, you know, in nature, this doesn't happen. You don't get SV40 causing tumors in hamsters, most likely. But it was an experimental trick that we used to understand why this viral proteins, why T antigens could transform cells. We infected the wrong hosts. However, we now know that even in the right host, a virus can cause transformation because we see rare spontaneous deletion of late genes in the right host. We see rare integration of oncogenes or T antigen genes in the right host. And I'll show you an example of that in a moment. So transformation is an epiphenomena of a unique reproductive cycle. It was also unique for RNA tumor viruses that have to integrate. That's a unique reproductive cycle. For DNA viruses, what is unique is they have to start the cell's synthetic machinery so that they can have their DNA reproduced. And so they have proteins called T antigens that turn on the cell cycle by inactivating RB and by inactivating P53. So when you block RB and you block P53, the cells divide forever. So as long as you have the T antigen present, they divide forever. They're transformed, and eventually they will sustain enough mutations to be a cancer cell. So they're on their way to becoming cancer cells. And the hash mark, just to remind you, RB and P53 are mutated in many different tumor types. So you can imagine they would be. They're key regulators of cell division. And in case you don't know what epiphenomena means, it's a secondary effect that arises from, but does not causally influence a process. So it arises from this unique replication, in this case, making a protein that kicks the cell into division, but it doesn't influence it. So whether a cell is transformed doesn't influence virus replication. Same for RNA tumor viruses. So now we can explain Henrietta Lacks's cervical tumor. So as you know, Henrietta Lacks died of a cervical cancer, but before she died, a piece of her tumor was taken out and put in culture, and that became HeLa cells. They are immortal. They divide forever. So in the 90s, the genome sequence of HeLa cells was done, and it showed chromosome 18, human papillomavirus, E6 and E7 genes are intact. None of the other viral genes are present. Remember, E6 degrades P53, E7 binds RB. You need just those two proteins to make the cells divide forever. So she was infected with a human papillomavirus. It's a sexually transmitted virus. It infected her epithelium, and just a very rare event happened. The genome was degraded, and the two genes, E6 and E7, integrated, and that made them transformed and divide forever, and eventually became a tumor. This integration is a rare event. Many, many people have HPV infections. And we'll see the numbers later when we talk about vaccines. But not everyone develops a cancer because you have to have this rare fragmentation of the genome and integration event happening. So here's the summary. We have our cell cycle there where the proto-oncogenes discovered by studying transforming retroviruses, they start the cell moving through mitosis. And then we have a stop signal at the bottom of the cycle, which is controlled by what we call tumor suppressor genes like RB. So the proto-oncogenes are dominant oncogene. You just need a mutation in one gene of the two copies in order to drive the cells into division. And the tumor suppressor genes are recessive. You have to inactivate both copies to allow the cell to pass through that checkpoint. And we identified the proto-oncogenes by studying RNA tumor viruses. We identified the DNA tumor virus, the tumor suppressor genes by studying DNA tumor viruses. I think this is an amazing example of how studying viruses that don't necessarily cause human cancers, but in animals, right? How studying that elucidates completely transformation and oncogenesis. So, you know, if anyone asks you, why do we study viruses that infect mice? You have a good answer for them. It illuminates many, many things. I say this because, I don't know if you remember, the governor of Alaska once ran for vice president. I forgot her name. Sarah Palin. And she was criticizing the NIH for funding research on worms and flies. And, you know, this is just ignorance because she didn't know, no one ever explained to her why this was important. But she was also using it as a political ploy. But this is another example. You know, what do we care about cancers in mice and hamsters? Well, this is why, because it illuminates how it works. Anyway, the amazing thing is that this starts with a tumor in a chicken. A chicken brought into an investigator who does some work, gets a virus, and then people start to study it and figure this out. So that's why this is a nice story to illustrate, bless you, to illustrate this history. Okay, next time we're going to talk about vaccines, how to prevent disease caused by virus infections.